All right, this is this is lecture 8A, part 2. We're continuing with renal system or renal physiology, and we went through the anatomy of the renal system. Just to remind you, this is a expansion of the cortex above these veins and arteries here, which here is, let me highlight it, above the corticomedullary junction. That's the junction between the cortex and the medulla. So outside of that, within what's called the cortex of the kidney, which is this area here, this outer area, that's where the glomeruli are located. There's the two glomeruli. This is the incoming afferent arteriole, then the glomerulus, which is a capillary bed, and then the efferent arteriole, which then goes on to form the peritubular capillaries all around the tubular duct system. There are two different types of nephrons shown here. One is a cortical nephron. That's the type that's typically found, but there are also some nephrons that are called juxtamedullary nephron. There's a cortical nephron, and these are juxtamedullary nephrons where the glomerulus is located closer to the corticomedullary junction. And the loop of Henle, this tubular loop going down, dives deeper into the medulla of the kidney, into that area of the kidney. And these are typically, these juxtamedullary nephrons are typically involved in forming more concentrated urine. Okay, and that was just a review. This is a high-powered magnification of the glomerulus here inside the glomerular capsule, or also called the Bowman's capsule, which is the beginning of the tubular system. Here's a afferent arteriole, and there's a efferent arteriole. I remind you that the tubular system is proximal convoluted tubule, then the loop of Henle, descending arm of the loop of Henle, then the ascending arm, and then the proximal convoluted tubule, and it always comes back close to the Bowman's capsule or glomerular capsule where it originated, and then it goes on, this is the distal convoluted tubule, and then it goes on to join here a what's called collecting duct. If we look at the high power magnification, you can see the part of the distal tubule, the distal convoluted tubule, that's coming back near the glomerular capsule, and in the wall it has a structure called the macula densa or macula densa cells. There are also specialized cells around the incoming afferent arteriole. These are juxtaglomerular cells. Together, the macula densa and the juxtaglomerular cells and the macula densa together form the juxtaglomerular apparatus. So these two, this and this, form the juxtaglomerular apparatus. Okay. And that, I believe, brings us to where I was before, where I ended off. So now we're going to talk about the physiology in the renal system, the actual mechanisms involved in urine formation. Essentially, forming urine involves three processes: filtration, reabsorption, and secretion. Filtration, of course, occurs here. The blood coming in to the glomerulus, that capillary bed inside the glomerular capsule, the blood in the glomerulus is filtered and the filtrate goes into the tubular system. It enters into the glomerular capsule and then goes into the proximal convoluted tubule. So that's filtration. And then, and that's A here, which is the same as 1. So that's the process of filtration. I really should have put this as A. This is, but it's defined here, so that's okay. The process of reabsorption is occurring in the tubular system, and it involves the reabsorption from the filtrate back into the bloodstream, in particular into the peritubular capillary bloodstream. So it's going back into the peritubular capillaries, reabsorption of certain components of that original filtrate. The other thing that happens is that there is also secretion from the circulation into the tubular system, and that's shown here, C. So that's the third process, C. Okay, so you have filtration and then reabsorption and secretion occurring. Now, as you know, hopefully, from what I said before, there are 2 capillary beds. One is glomerulus inside the glomerular capsule, and the second capillary bed are the peritubular capillaries around the tubules. The tubule starts with this glomerular capsule or Bowman's capsule, and then the fluid goes through it and out into the collecting duct as urine. But it takes quite a while for quite a bit of processing before that initial product of filtration becomes urine. And that processing involves reabsorption and secretion. So those are the 3 processes: filtration, reabsorption, secretion. The driving force in formation of a filtrate is the hydrostatic blood pressure. That's the main force. So water and small molecules, but not any significant amount of protein at all. Maybe a few small proteins do get filtered out of the glomerulus and into the glomerular capsule. Essentially, the filtrate is similar to plasma, but protein free. Again, there may be a few small proteins in there, but if they are, they'll get reabsorbed. So then in the tubular system, reabsorption and secretion of particular molecules, particular fractions of the filtrate occurs, and then the filtrate is in this way processed into urine. Here's an electron micrograph of a glomerulus. All this is glomerulus, and this is the area here is the showing the glomerular capsule. This is the beginning of the tubular system, and you can see that the proximal convoluted tubule is here. Okay, so filtrate is formed in this area. Sorry, in this area here, the filtrate is formed, and then it moves down the proximal convoluted tubule. Here what you see is that filtration is occurring because there are cells involved in that are wrapped around the outside of the glomerular capillaries, or the capillaries of the glomerulus. These cells actually are called podocytes, which you will see on, I think, the next slide. They're called podocytes because they have these foot-like extensions. Okay, so you have slits, filtration slits, that the filtrate coming from the space of the, from this space here, the space inside the capillary, so coming from the blood and exiting out, and it passes through these slits that are formed by the podocytes wrapped around the capillary of the glomerulus. And here you see some of these podocytes formed around it's an electron micrograph, scanning electron micrograph of the podocytes wrapped. Here, what you have here is all of this essentially is a single section of the capillary of the glomerulus. Here's another section here, and it's covered by these podocyte cells that have these extensions, foot-like extensions. That's why they're called podocytes. And little slits. You see how the little slits in there in between. All of this area here. All of this area here. In there you have these little slits formed by all of these foot-like extensions. Okay, in terms of comparative physiology, comparing between different types of vertebrate animals, only birds and mammals can produce a urine that is more concentrated than their own tissue fluid. So they can make a very concentrated excretion. I wouldn't call it urine exactly in birds, but the stuff they put out. Don't forget, birds only have one orifice or hole, and they do everything with that. So feces and uric acid and eggs and reproduction all through the same site. A little messy. So in mammals, the concentrating ability of the kidney is possible because of the anatomy, like everything really. The anatomy, the structure subserves function. Mammals do have a higher body temperature and higher metabolic rate, as also do birds. And in order to prevent a high rate of water loss, they have to be able to concentrate their urine and put it out in a quite concentrated form. Now, in humans, the proximal convoluted tubule is where 98% of the filtrate that's originally formed gets reabsorbed, at least in terms of many of the solutes that are in it and much of the water. So most solutes that get into that filtrate don't go out as urine, they're not in your urine, and most of the water that first forms that filtrate is not going to appear in your urine, even when you're putting out a fairly dilute urine because you're drinking a lot of water. You will not, most filtrate gets reabsorbed. The filtrate volume is fairly large. And most of that reabsorption process is occurring in the proximal convoluted tubule. Any glucose or very small proteins or amino acids will get reabsorbed out of the filtrate. Also sodium is reabsorbed, not entirely. It's a question of balance. The body is constantly maintaining a certain balance of sodium. Chloride typically follows the sodium, goes hand in hand with the sodium movement. So what you have essentially is that if you have a filtrate, most of the water is getting reabsorbed. Glucose, amino acids are being absorbed, reabsorbed. Sodium is reabsorbed depending on how much sodium the body needs, whether it needs to get rid of it or keep it. Now, you'll see later. Water is always moving typically by osmosis. anything that does get reabsorbed, of course, will get into the body from the filtrate in the tubular system, goes into the peritubular capillaries. Now, after the proximal convoluted tubule, you have the loop of Henle. And I remind you that that dives down into the medulla and then loops back up out of the medulla into the cortex and comes back near the original glomerular capsule where it was initially originated. When the fluid, which has been partially processed, that filtrate that's been partially processed, goes into the loop of Henle, it's still fairly similar to blood plasma in terms of its osmolarity. It'll have a close to 300 milliosmol. Now the thing about the loop of Henle is it creates a concentration gradient in the tissue around it, and this is actually called a countercurrent multiplier mechanism. What is happening is that as the urine flows down the descending loop, or the fluid that will become urine goes down the descending loop of Henle, it gets concentrated because water is reabsorbed out. Okay, water moves out of it, and then it becomes more and more concentrated till it gets to the bottom of the loop, and then it comes back up, at which point sodium is being reabsorbed, and so the osmolarity goes back to normal levels or even below normal levels. But this ability to move down the loop of Henle and become more and more concentrated and then move up the loop of Henle and become less concentrated is very important, and is shown. It's the counter-current multiplier mechanism or effect, and is shown here. So again, what you have is filtration occurring from the glomerulus into the glomerular capsule, and at that point the osmolarity is the same as plasma osmolarity of 300 milliosmols. And then it moves down through the proximal convoluted tubule, and most water and sodium chloride is being reabsorbed here in the proximal convoluted tubule. It then goes down the descending, let's get another color, down the, no, that's no good either. Okay, it then moves down the descending loop of Henle, the descending arm of the loop of Henle, and is becoming more and more concentrated. As you can see, it actually the maximum is 11 or 1200 milliosmols. The reason it's getting more concentrated is because water is moving out, and water is moving out by diffusion. And then it moves back up. Here you see 1200. That's the peak that it can reach. And then it moves up the ascending limb, and at that point the ascending limb is impermeable to water but permeable to sodium chloride. So sodium chloride can move out, and therefore the fluid inside, this fluid that was originally filtrated and is now being processed towards becoming urine, it drops in its osmolarity. So on the descending arm you have permeability to water but impermeable to sodium and chloride. On the ascending limb, that was the descending limb, and the ascending limb, and we'll talk about both thin and thick together, it's impermeable to water, both impermeable to water, but both are permeable to sodium and chloride. And so it arrives back here, and remember, they've moved these peritubular capillaries away from the tubules themselves just for clarity. But remember, all of the tubular structure is surrounded by peritubular capillaries, and the distal convoluted tubule here in this section here is always moving close to the original glomerulus where it originated. And it is there that some more water can be pulled out, So it arrives actually a little bit lower than normal tissue osmolarity of plasma osmolarity of 300, and then more water can be pulled out and goes to 300, and then it goes down the collecting duct. Now the collecting duct, if you're going to make a concentrated urine, the collecting duct is a site where more water is reabsorbed. But that really is dependent upon whether you do need to make a concentrated urine or a dilute urine. From moment to moment, depending on your condition, the amount of water and the amount of sodium that will get reabsorbed is dependent on what your status is. If you sit down and eat a whole bunch of chips with a lot of salt, then it'll trigger thirst because your plasma osmolarity will go up. It will it might cause some output of sodium out of your blood plasma into the loop of Henle. You will typically, where sodium goes, water follows. Okay, I'm sorry. You will lose salt, but try to retain your body. Your kidneys will try to put out salt, but retain fluid in order to get that osmolarity of the plasma back to a normal level. So you do have to always produce urine to get rid of waste solutes like excess nitrogen in the form of urea or excess sodium, but your body has to dissolve it in water. That's your solvent, and you need some water to carry it out. But how much water is being put out will vary depending upon your status, whether you need to keep more water in you or put more water out. If you need to keep more water in you because you have to lower your plasma osmolarity to a normal level. If it's a little bit high and you want to lower it to a normal level, you want to retain water. You don't want to lose it much. Or, for example, if you're somewhat dehydrated because of a lot of physical activity and not drinking enough water, then you'll tend to retain water in the kidneys, reabsorb more. You'll put out a more concentrated urine. And the example here on this slide is quite a concentrated urine, very concentrated. Urine, you can find urine ranging anywhere from 50 to 100 milliosmoles, to not 100, 1,200 milliosmoles. And I've seen this kind of range in the lab when we do urinalysis lab. But again, it's going to change moment to moment. If you're drinking more water, you'll probably put out, if you're drinking a lot of water, you'll put out more dilute urine. If you're not drinking enough water, if you have been active, you'll put out a more concentrated urine. It really depends on diet and the fluid intake. Okay, now as that filtrate forms and sodium and water are getting reabsorbed and it's going through here, the one thing that doesn't get reabsorbed along the way, at least through the initial part, the proximal convoluted tubule and the loop of Henle and the distal convoluted tubule, the thing that doesn't get reabsorbed is urea. Urea is our form of nitrogen waste. However, if the urea concentration gets really, really high, then some might get actually reabsorbed, but that's not a problem that you're reabsorbing a waste molecule, because it just gets secreted back into the loop of Henle. So what you're doing is you're putting out most of the urea most of the urea that initially goes into the tubular system is being put out as waste. Okay. And this is a diagram that sort of goes through the different steps. There you see blood in the system. This is supposed to be your heart here. So blood in the systemic circulation enters into the the capillaries of the glomerulus, and a filtrate is formed in the glomerular capsule and goes into the proximal convoluted tubule. Meanwhile, the blood leaving the glomerulus by the efferent arteriole is going towards the peritubular capillaries. So there's the fluid going down the tubular system, and the peritubular capillaries are wrapped around the tubules in order to allow for reabsorption and some secretion. And the fluid goes through the proximal convoluted tubule, the loop of Henle, the distal convoluted tubule, and gets processed, and then can still undergo further processing in the collecting ducts to form urine, and the urine gets put out in the end. I think that says urine down there at the very bottom. You can't quite see it. No, you can't see it. Okay, another somewhat animated slide here. This one is better. So blood goes in by the afferent arteriole, goes out by the efferent arteriole, into the peritubular capillaries, and then back to the systemic circulation, the veins, back to the heart. Blood plasma is being filtered in the glomerulus, and that filtrate is forming into the glomerular capsule or Bowman's capsule. Then enters the peritubular, then enters the proximal convoluted tubule. That area is the cortex. That's where the glomeruli are located in the kidneys. So all these glomeruli are located in the cortex. And that's the medulla, the outer medulla and the inner medulla. As you go deeper into the medulla, from the outer medulla into the inner medulla, the concentration, the osmolarity of the tissues is increasing because of that countercurrent multiplier mechanism. Okay, the convoluted tubule, or not convoluted tubule, the loop of Henle has an ascending limb, and which is permeable to sodium and chloride, but not permeable to water. And the descending limb is of the loop of Henle is permeable to water, but not sodium or chloride. The processed fluid, which initially was filtrate, then underwent reabsorption and secretion, goes into the distal convoluted tubule and then into the collecting duct, where it can further undergo further processing, where water can be further reabsorbed if you're going to be making a concentrated urine. If not, then water isn't reabsorbed in the collecting duct, and the urine goes out as quite dilute, quite a dilute fluid. And this shows the numbers, initially 300, and then it increases going along. It can peak at 1200 down at the bottom, and then it comes back up as the sodium and chloride are pulled out, and it can be lower than normal, 150, then some water gets pulled out, and in the collecting duct going down here, you can get a lot of water pulled out, so you can make a very concentrated urine up to 1200 milliosmoles at the maximum. Remember, this is the maximum concentration, 1200, right there. Doesn't show up no matter what color I make it, huh? Anyhow, that's it, 1200 right there. This is the same thing, and the numbers may not be exactly the same as the previous couple of slides, but the idea is the same. There's some range after all, but it's pretty much the same. this idea of varying permeability depending upon whether we're talking about the descending limb of the loop of Henle or the ascending limb, where the descending limb is permeable to water, not salts, NaCl rather, and the ascending limb is not permeable to water but is permeable to sodium and chloride.